Hey, good afternoon. I'm Dr. Joshua Green, and my patients always ask me about lab grown teeth. I get this question all the time. So I'm going to dive into the lit and try to bring everyone up to date. Now, it's an important topic for sure because tooth loss affects millions. People are living longer, medications dry out their mouth, so they're losing teeth. Partial edentulosum is on the rise. And the result is, how can they replace these inevitably missing teeth? Because there are issues with current solutions. Dental implants are great. You can't get cavities on them, but they can break and you can get periodontal disease around them. So you still have issues. Plus, they don't have any sensation, they don't feel pain. You can try to fix your teeth with crowns and root canals and fillings and whatnot, but even those solutions can run into problems, and there's a shelf life on many of them as well. So, what are we going to do? These limitations have really driven scientists to try to find a better solution. And that solution are bioengineered teeth. This is sort of the holy grail. And by the way, as a side note, some of the same people searching how to regrow teeth are also trying to regrow hair. So there's a lot of money and there's a lot of pressure to try to produce in this field. But the idea is that teeth are supported by nerves and have a blood supply through the dental pulp, and they can sense pain, they can sense pressure, and to an extent they can even repair themselves. If a cavity gets too close, they can start to do various types of eruption patterns. And now the idea is, and this is something that's said, this was a quote from the British Dental Association, that it's exciting, but it's a long way off. And I'm really have come to the same conclusion. There's some big issues with the types of approaches, but it may be that there's approach that isn't perfect, but is good enough and is already ready to implement. So the current estimates are 10 to 20 years, and that may even be conservative. So the first breakthrough, it really came on the scene back in 2009. This was a Kanaki Soju, who was able to harvest these germ cells. This was on a mouse model that normally develop into a tooth. And he was able to separate them, he and his team, into ectodermal and other types of stem cells, the mesoderm. So the ectoderm and the mesoderm are the two different types of cells. And they were able to mix them up and recombinate them. And they discovered that when those were reimplanted, it would create a tooth germ. They could recreate the tooth. However, these are the ideal situations where you're taking these stem cells that are already differentiated to form a tooth. So practically speaking, this isn't useful in a human model, but it does illustrate, this really set the foundation that this type of thing is possible. Now, the result is, in this mouse model, they were able to get a fully formed tooth, dentin, enamel, pulp, blood vessels, nerves. It could chew, it could feel pain. They were able to recreate this and essentially move it. So very exciting. They've tried various other approaches. This was a cell homing approach, and there's various other people. There's many, many teams working on these different approaches. But this was the idea that if you create a scaffold, which is roughly the shape of a highly immature tooth germ or tooth bud, and place that in an animal or even a human, you could put growth factors in it that would then cause stem cells to start to migrate into that matrix. And there are stem cells, there's mesenchymal stem cells in bone marrow, like the kind in your jaw. So these stem cells are there. And if they're in the right situation, they can begin to differentiate into the types of tissues that are present within our teeth. The real problem with this is it's very, very difficult to get it to form enamel. That's made by ameloblast cells, and those are specially differentiated ectoderm cells. An interesting pulp, dental pulp and various other things are already ectoderm, or gum tissue, are already ectoderm. So you can use other types of sources for that, but it's difficult to get a perfectly formed tooth using this method. And that's really where we've been working ever since. We've tried to use human tooth bud with readily available human cells. But again, you can use adult gum tissue and then the mouse embryonic cells, those germ cells. It's very hard to get human germ cells. And this was successful. It was able to develop a small tooth. However, a lot of these ones, they can even use urine cells. So there's all kinds of different ways to generate stem cell lines. But once again, these various, so it's often end up not being hard enough or the wrong shape. The real problem is a tooth is incredibly complicated. It's a mini organ with several different types of tissues where the enamel actually grows outwards and the pulp grows back the other way. So again, getting it to develop in a logical way is very, very difficult. Enamel is the outer hard layer, which is heavily mineralized. It's a cellular. Once it's completely done, there's dentin underneath, which is much softer. It has dentinal tubules, and finally there's pulp, which are the which contains predentin, various the mechanisms to make more dentin, and it can still grow inwards, and it has nerves and blood vessels and even immune cells and stuff like that. Finally, there's the periodontal ligament, which is a fur that basically goes all the way around the tooth, which is then surrounded in cementum, which is around the dentin, and it goes inside that and also connects onto bone. And that's what provides the shock absorber that allows teeth to stay in our skeleton. It's very, very strong. It works very well and gives you some proprioception when you push on the tooth. So there's a lot going on, and all of that is involved as the tooth erupts into the dentinal cavity. So trying to recreate all of this in a way that makes sense requires very precise timing. Different things happen. And as a result, it's very hard to create a matrix that's artificially made that does all of these things. So a lot of what they've done, there's a team at Tufts that has come remarkably close by bioengineering teeth in pig jaws being roughly the same size as ours, the teeth being fairly similar in size. But what they had to do is use a decellularized germ from the pig jaw. So they're able to take that out, but in a way that's sort of cheating because it has all the information already set. It's got the structure, the matrix already set, and they were able to place human cells in that. So is it possible they could transplant that? We actually transplant pig bone, pig tissues, pig hearts, all kinds of stuff, or parts of pig hearts into humans all the time. Is it possible we could take a decellularized germ from a pig and put it in our jaw? And I would say that's extremely possible because we do it very often, as long as it had the right growth factors. So again, there are future directions here. But right now, obviously, a human jaw, if you're trying to regrow a tooth in a human jaw, you're not going to have a tooth germ handy. So you have to find another solution. Now, the scaffold, this is nature's template. And the idea, again, what she did is she removed all the cells from the tooth bud, leaving only that matrix. And that matrix is really what we would need to copy. And they were able to reseed that with tooth bud cells, human stem cells. So your ectodermal cells, your mesoderm cells, growth factors, and it's incredibly complicated. And they were able to make that grow into a fully functional tooth. And this preserves those natural cues. But how do we get this in a human? And once again, the ethical thing is human embryonic tooth bud cells are very hard to get. ethically. You can't really donate that kind of stuff. People use them. So the idea is the problem, once again, is recreating the types of signals are incredibly complex. If you were to remake this very complicated tooth structure, you would have to really, we would have to go a long way before we actually get there. And there's been some bright spots, but still, in a way, we're sort of cheating. We're not doing this from scratch yet. Now, there's a very there are breakthroughs. There's many people working at this. King's College in London, they're making materials that are working on this. And there's been a lot of progress on making these types of artificial matrices. They can be made out of gel, cellulose, whatever it is, some sort of thing that can resorb easily. Now, by the way, these still need to be surgically implanted. You can't just put it on the outside and expect it to grow. You have to actually put these in there some way. So again, it's still a surgical procedure. So it really has a long way to go to overtake the dental implant. And again, these mimic the natural cell line, but again, they're not perfect. But I think, in my opinion, it's very possible there's going to be some sort of happy accident for this, because already they're noticing that sometimes they accidentally make bone-like structures. And even if that's all we end up doing is just creating a bone-like structure, that will be incredibly powerful for grafting, even if we eventually do an implant. So it's important. So really, there's a couple of different options. Maybe it's possible eventually to be able to grow a tooth in the lab and then transplant it. We may be able to grow it in the body and use our own stem cells. And I think that would be the gold standard here. But really, there's a bunch of different approaches, a bunch of different ideas, and nothing is quite worked yet. There is a long way to go. Now, there's a third idea that's out there. And this is what people tend to hear about on the news, these regrowing things. This is actually human trials in Japan. Very exciting stuff. But there's something called USAG1, and it is a brake on tooth formation. And what's exciting about that is that we actually have the potential to make a third set of teeth, or perhaps even more than that. And this USAG1 is a chemical that stops tooth development. And by creating an antibody to it, they're able to essentially suppress it. And when that happens, at least in animal models, those models start developing new teeth. And this is very useful because there are animal models that don't make teeth. They're missing teeth. There's humans that also miss many teeth, and they are able to generate another set of teeth. The problem is it's not exact, where you may generate a tooth in an area that you already have a tooth. You don't generate every tooth. Younger people tend to generate more than older people. And obviously we'd be looking at older people that need a full set of teeth. And no, at this time, you may get a few new teeth, which may be good, but you're not going to regenerate your whole dentition. So there are some, and maybe you'll be able to isolate it, but there's a lot of work to do. It's important to explore, and it's non-surgical, which is great. And it's something to monitor. I think this is an exciting medication to watch. It's actually got important for kidneys as well. So again, very exciting, but it's not really targeted to grow a single tooth, which is what most people need right now. Once again, the enamel challenge, the holy grail of tooth remineralization, because it goes from a different type of tissue, it requires a highly specialized structure to grow. It's growing in a different direction and from a different tissue. So even if you figure out the mesenchymal tissue, you can grow the entire tooth and you still can have issues with the enamel. Now, for me as a dentist, I do a lot of crowns. So I don't see a problem if you develop a tooth that's got no enamel. I can just crown that tooth later, and then that will essentially replace all the enamel on the tooth. So it may not be essential, but still in a perfect world, that's exactly what you would want. And again, you can't. Once the tooth's developed, you can't develop any more of it. Those ameloblast cells are gone from the body. They can't be found anywhere. So once again, this is why cavities develop permanent damage. But even if we don't develop the entire tooth, there are attempts to develop or help do different segments of it. And one is enamel, cordon, actually. And what that is a simplified version of the protein that it's amelogenin, which is incredibly complicated natural protein. So they knew they could never make that, but they were able to make a much more simplified protein that can create similar structures. And that's exactly what this does. So it does a peptide gel and it simulates mineral deposition. So it takes some of the minerals already found in your saliva, in your mouth, and creates an enamel-like substance. Synthetic enamel, if you will. But obviously the issue is that it doesn't make a particularly thick layer. We're only talking about, a couple of micrometers. It's thin, it's very liquid-like. So my hope is eventually it will be vastly improved and you'll be able to replace 1 millimeter or 2 of enamel. But again, at this particular time, still composite and fillings and stuff like that are required. But it does have potential and it's very exciting to see. Now, anytime you're talking about incredible complexity, obviously machine learning, all these different types of things are incredibly influential in genetics, in medicine, protein folding. They can simply solve issues that you really can't solve by hand. So it's exciting, especially when you have a problem, a 3D problem of how you create a certain shape of a tooth that happens to be something that's already being studied in other aspects, because there's a big business in generating dental crowns. Is there a way to also engineer a dental crown based on the types of growth factors that would require to make that crown? Well, it's very possible. Can you create a serum? Can you help design a scaffold for a certain cell? Can they do explorations of different proteins and model these types of things? It may be an important tool. And again, a lot of these things are already being discussed, particularly the idea of creating a scaffold is really the problem right now. Are there different approaches? So it remains to be seen. Now, again, it's very exciting with this new drug in human trials in Japan, the USAG1 we're talking about. Even though that would only be at this time, it's only if you're missing multiple teeth and you're young. Most likely you had a developmental condition, of which there are some where you have a potential to grow new teeth. However, once again, you could grow a tooth in a place you already have a tooth, which could potentially cause you issues. So it's got a long way to go. But right now we have different bits of pieces. We figured out how to grow ectodermal cells, and we figured out how to grow dental pulps and various other mesenchymal cells. So if we could get everything all together, or we could use various parts of an animal model, or create a tooth that's just good enough that you could crown it and figure it out the rest of the way, this could be used. But at this time, there's really nothing on the horizon that's sophisticated enough to get a tooth that is at least developed enough to consistently go in a human. A lot of them are getting stuck at an early stage, and it's just not there yet. But, so 2035, 2045, I think as long as there's a continued effort for it, this could certainly happen. I think it's on the horizon. I think it will happen. We just need to figure out, the right recipe to make this work. So who's going to benefit first? Obviously right now we're looking at people that kids, younger people that don't have teeth. And Eventually, adults with single tooth loss. The reality is dental implants, which are titanium, are very good. Most options on the horizon for will also require a surgery. So the benefits are somewhat minimal. So you would have to have them be incredibly good or not be a candidate for a dental implant for this to really start to make sense. Um, so obviously there's a lot of implant patients out there, and there's various things you can do with dental implants that you can't easily do otherwise. But the idea is that this certainly has potential and could be done in certain areas where you need to regrow bone and regrow soft tissue, which you can't easily do with dental implants right now. And that's really the problem with dental implants. If you have plenty of bone and plenty of soft tissue, implants are perfect. But if you don't have those things, it's going to be a real struggle for you. And that's the crux of the problem with implants right now. So the changing role of dentists, if this becomes 20 years, which is something I may see in my career, I hope I do, is all of a sudden your role is going to be really understanding how this works and possibly implanting a tooth blood or a scaffold instead of an implant and letting it take hold. You may have to wait for the teeth to grow. Implants often are not immediate, by the way. I usually wait 4 months. A tooth that grows may take slightly longer than that. And of course, the way we monitor these is going to be different. The way they integrate, managing the eruption, a lot of the same issues that could occur on the tooth they just lost could probably reoccur on their new tooth. And there may be a lot of these traditional techniques. You would still need to maintain these new teeth. So the idea is you may start regrowing teeth and managing that process, which would be very exciting. I think we'd get some really good results for people. And, just to reiterate, Takahashi's breakthrough, USAG1, very exciting, very flashy. They were growing a lot of teeth out there, but again, it's not zeroed in. They are looking at 30 adult volunteers that are missing teeth. If you're a candidate, which most people probably are not, this could be wonderful. There's also artificial tooth matrices, very exciting, but at this time, it's still not sophisticated enough to consistently make a big, beautiful tooth every time, but they are working on it, being able to differentiate what tooth is actually being developed, what kind of cusps, but you need to, again, be highly, highly specialized in this. So there's a lot of ideas. Do we generate the tooth in the lab? Do we use a scaffold? Do we use a pharmacological, like a USAG1 based approach? Or maybe we start just targeting pulps. There's various techniques where you could possibly, instead of doing a root canal, what if you were just able to regrow the whole nerve, sterilize the whole tooth, and then try to regrow the whole nerve? This is actually already done on pediatric patients. Revascularization has been done for a long time. So it's not out there. It is out there. Regrowing the enamel, we already talked about. Regrowing the PDL, very complex. That's a tough one. That's the holy grail of periodontics is regrowing the PDL after you've lost bone. Can you regrow that whole apparatus all at once? And it may be challenging, but even being able to regrow a small part of the tooth may be much better than a filling or some of the other options we have right now. And, really the idea is that you could possibly just take a sample of cells from your gum, which have the ability to form these ectodermal layers. You may be able to have mesenchymal cells from your jawbone migrate towards that tooth bud, and then your whole tooth would grow. And they figured out little bits and pieces of the puzzle, but nobody's quite put it all together yet. And that is something that could be done. I think it's on the horizon. and will happen soon. So either way, hope you guys enjoyed this. We'll be talking about this again. I'll be back again tomorrow in the morning at 6 a.m. live. So either way, thank you very much.